going to go first. Should I? You think I should send him a WhatsApp about the link?
No, I'm pretty sure he's chairing a syndicates meeting, I think, at the moment. So I'm sure he'll be able to give you a call.
And tie for everything you do, even if you sell newspapers.
So I did my postgraduate training in the UK too, and that's probably why I reverted so desperately to the other extreme.
I did some of mine in the US, and they're not that particular, but they like a tie at least.
Yeah. Carl, are you going to be sharing your screen at any time? You're going to be sharing slides?
Yeah. I mean, what do you want me to do? You want me to share them?
Can you just do that, test that quickly now, just to make sure that everything's working?
Okay, share screen.
We can hear you. Can hear you.
behalf of the editorial team and as part of the journal's educational endeavor, I welcome everyone today to our second webinar on COVID-19. The timing of today's webinar could not have been better, coinciding with the arrival of the first batch of vaccines into South Africa and the release of the results of the J&J vaccine last weekend. So firstly, I would like to welcome our esteemed panel for our discussion this afternoon. I'm sure it will be a very lively discussion. Just before we start, some information. Following the panel presentation, there'll be a Q&A. A recorded version of the webinar will be available, and the link will also be published in the next issue of the journal, which comes out in the first week of March. Please feel free to add your comments and questions in the chat window. We will be monitoring this, and we will try our best to incorporate as many of the questions and comments in your Q&A session at the end. So to kick off the webinar will be Professor Penny Moore, who is the Department of Science and Technology and the NRF Saatchi Chair of Virus Dynamics at Wits, and a senior scientist at the National Institute of Communicable Diseases. Professor Moore is a PhD in virology, and she has been actively studying the COVID variants in circulation in South Africa. And I really can't think of anyone better to educate and update us on the dynamics of the COVID-19 variants in South Africa. Welcome, Penny, up to you.
Thanks, Praveen. Let me see if I can share my screen. The first hurdle always. Is that up and running?
Yep.
Cool. Yeah, thanks very much for the opportunity to come and talk to you all. So you've got three speakers, as you've all gathered today, and I was tasked with giving you some context into the new variant in South Africa, variants elsewhere, and why they arise and why they matter. So let me start by saying that it's not a surprise that the SARS-CoV-2 virus that we have in South Africa is mutated. Mutations in SARS-CoV-2 occur constantly, as is expected for an RNA virus. And this phylogenetic tree on the left is put there to give you some sort of sense of the level of mutation in SARS-CoV-2. So each of these unreadable numbers down the left-hand side is a sequence that's been obtained from SARS-CoV-2, and you can see that they all differ subtly from one another. So it's not a surprise that there are mutations arising. Compared to viruses such as HIV, SARS-CoV-2 actually mutates relatively slowly. But there's a continual arms race between virus and host in all cases, and this is no different for SARS-CoV-2, that results in the selection of beneficial mutations. So mutations arise, some of them confer a benefit somehow to the virus, and that can occur in many different ways. And those particular mutations are selected for by the virus because it gives some sort of advantage. And those then propagate to varying degrees across regions, and then in some cases across the world. In the case of SARS-CoV-2, the fact that we've had so many millions of people infected essentially means that the virus has really had every opportunity it needed to become fitter, however you define that. And I'll get into that a little bit later. So we've heard much discussion in the last weeks and months about variants of concern. Variants of concern are named because they are concerning for several reasons. The three main variants that you would have heard about are the variant that was first detected in the UK, variably called B117 or 501YB1, and they're two different systems of nomenclature, and it does add a lot to the confusion. So bear with me, I will use this particular name as I go through the talk, 501YB1 for variant 1. The lineage that you've heard a lot about in South Africa is 501YV2, otherwise known as B1351. And then more recently, and I'll mention this briefly at the end, there's been the description in the press of a new variant that's emerged in Brazil called P1 or B501YB3. You have to wrap your head around these numbers. So I thought I'd start by explaining, because I think this is also an area that's sometimes confusing, what the difference is between a mutant, and as I said, mutants arise the whole time, and a lineage. So when we talk about a new lineage, we're talking about clusters of or families of related viruses that share some particular feature that clusters them together and differentiates them from other viruses. So Lineage is not one single virus. It's a number of related viruses, and that's shown here in the first description that was ever made of the new variant that was first detected in South Africa. This is a paper from Tulio D'Oliveira and colleagues that's on medRxiv. And I've put it up here first to explain what we mean by lineage. So 501YB2, the South African lineage, is all of these yellow dots over here. And you can see that they cluster on the phylogenetic tree very separately from previously isolated South African viruses shown across the lineage in red, and from other global viruses. But each one of these is a separate sequence. So to make the point that there isn't one 501YB2, it's a family of related viruses. So to take a step back and think about where 501YB2 came from, as you'll all be aware, there was an early and very rapid resurgence of SARS-CoV-2 in October, the beginning of the second wave. And that resulted in really intensified genomic surveillance. So we've always had in South Africa, since the beginning of the SARS-CoV-2 epidemic, we've had good sequence coverage, and that's through a consortium of sequencers across South Africa that have really worked very closely together to generate good sequence coverage across South Africa. In many respects, we're one of the leaders in this region. But they identified 501YB2, and they tracked it over time, and they showed that by mid-December, and you can see here in yellow over time, 501YB2 had come to dominate the South African pandemic. And now as people sequence isolates from across South Africa, somewhere in the region of more than 90% of new sequences that emerge from people newly infected with SARS-CoV-2 are the 501YB2 strain. So it's really swept from the Eastern Cape, where it was from the Eastern Cape across South Africa, and indeed across the world. So there is this notion that viruses can be contained to countries, and you will all be aware of the travel limitations that have been imposed in some cases. But viruses do travel with people who travel, and the 501YB2 lineage has now been detected in several countries, as you can see here. So not in many cases, not very many. So what this light pink here means one confirmed case in the country, but clearly spreading across the globe. Of course, it's not the first lineage that we've heard a lot about in the press. So the first lineage that was actually described as the 501YB2 that was first picked up in the UK. And that was a consequence of the same huge increase in cases in the UK, and the subsequent identification of a lineage that has a mutation at position 501. And I really wanted to make the point because I think people tend often to assume that the South African variant and the UK lineage are the same. They're not. There's only one common spike mutation, and I'll explain what spike is just now, between those two, which is a mutation called N501Y. And that's important because it confers increased binding to ACE2. So you will have heard that, and I thought I'd better explain what that meant. So ACE2 is the receptor on the human cells that SARS-CoV-2 binds to infect cells. So the 501Y mutation that sits here at the interface between the viral spike protein and the human receptor increases affinity for that. And that is the reason, we assume that is the reason, that this 501Y mutation has been associated with increased transmissibility in the UK. There is an assumption in many cases that because we have in the South African variant also got a 501Y mutation, that for example, the huge second spike that we've been dealing with is a consequence of increased transmissibility. But actually, I'd say the jury's still out on that. And I'll explain again a little later why that is. But viruses are complex things. They don't rely only on a single mutation. Of course, I mentioned a third variant, 501YB3. This is a new variant that more recently described coming out in Brazil, the Brazilian variant. But this is not the end of variants. And I think it's something that we all have to accept is going to be a constant part of dealing with this coronavirus pandemic. As these viruses emerge and as they are dealing with different pressures in different populations and dealing with immune responses and dealing with vaccines and dealing with host cell pressures, they're going to change. This is what viruses do. They mutate, good mutations are selected for and they get better. But beyond transmissibility, which I've already mentioned, why is there such particular concern over the variant that was first detected in South Africa? And I'm going to show you on this model of a spike protein why we're so concerned. So the spike protein is it sits on the outside of the viral particles and forms the crown or the corona that gives the coronaviruses their name. So this is the protein that is responsible for binding to the host cell receptor. Binding enables viral infection. Because it's also the predominant way for the virus to be, well, the only way for the virus to be able to get into host cells, antibodies that can prevent that from happening are clearly very important. And those are what we call neutralizing antibodies. Neutralizing antibodies are able to bind to the spike and stop the viral particle from being able to infect host cells. So this is very much the target of the immune response, and it's also very much the target of vaccine design for that reason. It's a very complex protein, and I don't need to go into it in particular detail, but what I want to show you is where the mutations that define the South African variant are located and why it matters so much. So there are two regions that are of particular focus for vaccinologists and virologists. One of these is called the receptor binding domain, or the RBD you would have heard about. And the RBD is the particular part of the spike that is responsible for binding to ACE2. And I've already mentioned the 501 mutation that enhances affinity for the receptor, and it sits up here. But the South African variant also has two other mutations at positions 417 and 484, and they cluster together here. The second kind of set of mutations here are clustered in the cyan part of the spike, which is referred to as the N-terminal domain. And you can see there's a whole bunch of mutations, and these little red dots indicate mutations, and even a deletion where the virus has essentially chucked out 3 amino acids from this region. And anytime you see clustering like this of mutations, it suggests that there is a functional consequence to those. And what's particularly worrying in this case is that these two bits, the N-terminal domain and the receptor binding domain, are the precise bits of the spike that the immune system preferentially targets. So antibodies that are directed at this protein and are able to neutralize almost exclusively bind to these two regions. So it's very suggestive that this virus has accumulated these mutations in order to escape immunity. So I'm going to show you a little bit of data now from our group that confirms that these mutations do in fact mediate immune escape. I'm going to start with monoclonal antibodies, and the reason we studied monoclonal antibodies is twofold. Monoclonal antibodies give very nice clean results, so it's very easy to understand what you're dealing with. But monoclonal antibodies have also, in the case of SARS-CoV-2, become very important from a therapeutic and a prevention point of view. Perhaps not so much in South Africa, where it's not likely that we're going to receive monoclonal antibodies anytime soon. But certainly in the US and elsewhere, there's a large focus on these monoclonal antibodies. And there are kind of two classes of them, and I'm not going to get into the specifics, but the class 1 and class 2 neutralizing antibodies. Class 1 neutralizing antibodies bind specifically to position 417. And you remember that that's one of the sites that's mutated in 501YB2. The other class of antibodies, inconveniently, also bind to one of the mutated sites in SARS-CoV-2, position 484. So that was immediately suggestive that these two mutations had been incorporated into the virus to evade class 1 and class 2 neutralizing antibodies. And indeed, when we tested that, that's exactly what's happened. We tested first a series of 3 class 1 antibodies that rely on this K417 residue. And you can see that they were completely knocked out in their ability to bind to the virus. So black here is the wild type virus, red is the 501YB2, and you can see they're no longer able to bind. And we also have data showing that they're no longer able to neutralize the virus either. And the same is true for the class 2 antibodies. Those are the ones that rely on 484. And again, you can see that these 3 members of that class of antibodies are no longer able to bind. So essentially 501YB2 is resistant to many, and I'm showing you here a subset of data, to many therapeutically relevant monoclonal antibodies. But as I said, the monoclonal antibodies are unlikely to come to South Africa, and we're probably more interested in what happens in what's called polyclonal plasma, so essentially blood from people, which contains many different types of antibodies that recognize many different parts of the spike protein. So we did essentially the same experiment here. We studied blood from 44 individuals who had previously had COVID-19 and asked whether the antibodies were still able to maintain activity against 501YB2. So over here, you can see at the bottom here is the original virus, the 614G. That's the kind of wild type virus previously circulating in South Africa. Here, we tested them against a virus that contained only those 3 mutations that are in the RBD. So it's positions 501, 417, and 484, just those 3 mutations. And here's the full 501YB2. I just want to point out that this is a logarithmic scale. So it's, when you see a decrease here, it's a big deal. And you can see that all of the 44 individuals that were previously showed a range of activities from up to 1 in 16,000 down to 1 in 200 or 300, 1 in 50. Almost all of them show a downward trajectory when tested against the RBD variant, and a further downward trajectory when tested against 501YB2. So they'll all lose activity, in some cases substantial activity, as I'll show you on the next slide, against the new variant. So this is kind of a simpler way of showing this data. Here's data against the original virus. I'm sorry, I've lost the key, but the dark blue means fairly potent and the light blue means less potent. Against the RBD only, 27% of them are no longer able to neutralize that virus, and half of them are no longer able to neutralize the 501YB2 variant. And as you can imagine, this has pretty significant implications, both for reinfection and for vaccine efficacy, which I'm sure that Professor Marley is going to talk about much more in his talk, which follows mine. We're not the only lab to have shown this. Happily, the data has been corroborated in a live virus neutralization assay by Professor Alex Segal at CRISP and Ari. Same data here, you can see that there's pretty good activity against using plasma from the first wave, but there's almost no activity left against 501YB2 viruses, ranging here from a knockout to a kind of a 30 or 31-fold reduction in activity against the new variant. So it's clear that 501YB2 has picked up a series of very useful mutations. I didn't show you the data for the NTD, the cyan part of the spike, but the 501YB2 has managed to evade antibodies to that site. What I've shown you shows that it's managed to evade antibodies to the RBD, which is really the major focus of the immune system. And I think what's important to point out is that this is becoming, these particular mutations are becoming a common pattern. So I mentioned the 3 major lineages that have been described. And if you think about and look carefully at the mutations, you'll see that they are common. So all 3 of these lineages contain a mutation at 501. Our virus that was first detected in South Africa has a 417 and a 484 mutation. The Brazilian strain has a 417 and 484 mutation. And now within the UK, they're starting to pick up viruses within the B501YB1 lineage that also contain mutations at position 484. So there is what we would call convergent evolution, which I think is suggestive of the fact that the virus is dealing with virtually the same sort of immune response in infected and vaccinated people. And it's taking the most obvious and shortest route for that particular virus to become resistant to a very common class of immune response. I think it really highlights the need for continuing genomic surveillance linked to lab studies to define what the importance of all of these mutations are. And we're in a good position to continue doing that in South Africa, but it certainly becomes very important in other countries as well. So I'm going to stop there. I hope I've persuaded you that there are many, many lineages of SARS-CoV-2 emerging globally, often with convergent evolution. In other words, they're developing the same mutations, and we need to track them very carefully. We need to understand, and we need to plan for them in our next vaccines. This is not a once-off event. The 501YB2 lineage, first described here, completely escapes 3 classes of monoclonal antibodies. And I think more importantly for us, shows substantial or complete escape from neutralizing antibodies in people previously infected with SARS-CoV-2. And it does raise the prospect of reinfection with antigenically distinct variants. So people who were infected prior to the emergence of 501YB2 may be very susceptible to infection by the new variant, more susceptible than they would have been to the same variant. And I think it provides a clear mechanism for the reduced efficacy of spike-based vaccines that you will have heard a lot about in the last couple of days, as Praveen mentioned in the news, and as I'm sure Shabir is going to tell you in his talk. And so I will stop there and see if I can work out how to get out of there, and hand over to Professor Mardi.
Thanks, Penny. Thanks for those insights, and especially about the neutralizing antibodies with the South African variant. So I think it's ideal that Professor Shabir Mardi comes in. He's probably well known to most of the audience. He has just assumed the role of deanship at the Faculty of Health Sciences at Wits, and he's also Professor of Vaccinology at Wits. Welcome, Shabir. He has a very deep knowledge base on most aspects of vaccination and has been a principal investigator on many vaccine trials in this country. So Shabir will lead on where Penny has left off, and hopefully we'll discuss the current science of the COVID-19 vaccines, hopefully it will help dispel some of the false mythology around the vaccines. Shabir, welcome.
Thanks, Praveen, and good afternoon, everyone. Just give me 1 second. Okay, so thanks for the invite to chat to you about where we are with COVID-19 vaccines. And I think a good starting point is just to reflect exactly where we are in terms of the trajectory of the resurgence that we experience in South Africa. This is a bit of a dated slide. I think it's about 2 weeks behind. And the most important message. Pardon?
I think we can't see your slides. We're seeing the other screen, unless it's just me.
You think? Okay, let me sort this out. Sorry. So you can't see my slide.
And I can see a nice sunset.
Okay, my apologies. Can you see it now?
Yes.
Yes.
Okay, my apologies. Okay, so like I said, I think it's useful for us to just reflect in terms of where we are with the epidemic curve in South Africa. And this is a bit of a dated slide, about a week old. But I think the important message here is twofold. And also it sort of assists in terms of explaining why we've experienced what we have in South Africa in terms of the resurgence, as well as possibly explaining the reason why the variant arose in South Africa, which seems to be resistant to both natural induced immunity, as Penny had indicated, and there might be also some issues in terms of vaccine-induced immunity and the resistance of the virus to the killing activity of that antibody, which is induced. So where we are in the resurgence is we are second wave, which for all intents and purposes has exceeded the first wave, both in terms of the number of cases as well as in terms of the number of deaths. So based on the MRC excess mortality data, during the course of the first wave, we settled around about 40 to 45,000 deaths having occurred from COVID-19, and that is using the excess mortality data. When we look at reported COVID-19 deaths, it's about 50% of that. Currently, the reported COVID-19 deaths is in the region of about 45,000 for South Africa, but the excess mortality data, which is generated by the MRC, indicates that already about 2 weeks ago, we were sitting at 125,000 deaths that were most likely attributable to COVID-19. Now, what we also experienced during the first course of the first wave is a tremendous force of infection in many major urban areas. One of the early sero surveys that was done opportunistically on people living with HIV and women attending antenatal clinics in the Western Cape, in the Cape Metro, indicated that anything between 35 to 45% of individuals might have been infected during the course of the first wave. We have subsequently just completed a sero survey at a household level in Gauteng, and the average across the survey is about 25% seropositivity after the first wave. And I'm sure as we dissect that by different sub-districts, we probably will find a range of anything from 15% in an area such as Centon, as an example, to as high as 40 to 45% in neighboring Alexandra. So why is this important? For all intents and purposes, viruses mutate sometimes purely by accident, but usually viruses mutate as well as bacteria. They usually end up mutating because they're under some sort of immunological pressure. And it's that immunological pressure which needs them to mutate to survive and continue propagating. And what we see happening in South Africa, as well as what we see happening in Brazil, in Manaus, which is another setting where during the course of the first wave, it's estimated that as many as 70% of people in Manaus had been infected. In these two settings, we see the evolution of the variant that Penny had described, which essentially seems to be resistant to antibody that is induced following natural infection by past variants of the virus. And that is because of mutations at very critical components of the genome that codes for specific epitopes against which the antibody usually works to reduce activity. So, like I said, the resurgence that we experience in South Africa, another important aspect is that in the context of having had 30% of people in South Africa, especially in urban areas, likely having been infected, in terms of the immunology and biology, if this infection had actually induced some level of protection, the resurgence that we experienced should probably have been less severe than what we experienced the first time, because we would have had a sufficient percentage or a large percentage of the population that have developed immunity, and consequently they should have derived some level of protection against reinfection. Whether they derive protection against severe disease but not against mild and moderate infection, that is another issue. So in South Africa, the timing of the resurgence coincided with the identification of the B1351 variant, which Penny has described in detail. And as we know now, the variant was first identified in the Eastern Cape in response to the resurgence, which first started in the Eastern Cape, and subsequently that variant has spread throughout the country. Currently, more than 90% of all infections that are taking place in South Africa are because of the variant. Now, these sort of mutations that take place, again, to emphasize, it's common for all viruses. It's not unique to SARS-CoV-2. But for all intents and purposes, most people probably expected a much slower rate of mutation taking place with this virus than, for as an example, an HIV virus. But that probably hasn't turned out to be the case. It seems that critical mutations of this virus probably has caught many people by surprise. And that's why there are a huge number of issues that have been raised in terms of exactly whether the vaccine efficacy that's been reported from B1351 variants would be as good as against the B1351 variant. So understanding these variants is important both from an epidemiological perspective in terms of understanding whether individuals that have been previously infected are at risk of being reinfected after having induced natural immunity. Like I mentioned, it's got implications in terms of vaccines. Today, we've just seen reported that some monoclonal antibodies have now been shown to fail in terms of management for the variant that's circulating. And then it's also got implications for diagnostics. As an example, some of our PCR tests are not based on a nuclear protein, but are rather based on a spike protein, which targets these sort of components of the protein for which mutations have occurred. So it might actually reduce the sensitivity of some of the diagnostics. And it's got implications in terms of vaccine rollout. Now, I think Penny covered this. I'm not going to go into too much detail. The big concerns about the variant are really twofold. The one is related to the N501Y mutation, which has been shown to increase the affinity of the virus to bind to ACE2 receptors and consequently increases the transmissibility of the virus. In the United Kingdom, it's been suggested that this virus with this mutation is probably at least 53% more transmissible than earlier virus that was circulating. And in fact, there's just a report today from the United Kingdom, which suggests that the virus might also be more virulent, in that it seems the mortality due to this virus attributable to this variant probably is as high as 60% more than previously, which wasn't data that was previously evident. We're not too sure in South Africa. It seems that the case fatality rate, at least for hospitalized patients, has remained somewhat stable compared to what we experienced the first time around, so we're not too sure. But as Penny pointed out, this variant that's affecting South Africa, as well as a variant that is now evolved in Brazil, and as of yesterday, modification of the N501V1 variant in the UK. These variants also include mutations of the RBD, the receptor binding domain. And I'm not going to go into the detail except to say that these mutations, additional mutations involving the receptor binding domain and terminal domain, are important in a context that we know that antibody that is actually raised targeting those specific epitopes of the protein are extremely useful in terms of neutralizing the activity of the virus. And much of the vaccine, in fact, all of the vaccines, besides the inactivated whole cell vaccines, currently have been designed based on a prototype virus. So that whether it's the messenger RNA-based vaccines or whether it's a protein-based vaccine or the vector-based vaccine, they're all designed to induce antibody against these epitopes, but in relation to the prototype virus rather than against specifically to the variant that's circulating. So Penny mentioned the issue of the B117 mutant, as she said that now this mutant has actually evolved even further, and it now has got an E484 mutation, which means that it probably will have reduced sensitivity both to past infection as well as vaccine-induced immunity. And similarly, the variant that arose in Manaus in Brazil, which seems to be a completely different lineage, has got these critical mutations, which Penny has probably described earlier and which I've just gone through. So what is the implications of all of this for vaccines? And we're fortunate in that we've now conducted 3 vaccine trials, and inadvertently, what ended up happening in South Africa is that the efficacy of these vaccines, because of the timing of enrollment of the participants, has resulted in these efficacy estimates being in relation to how well these vaccines work against the variant, and not anything about how well it works against earlier prototype virus that was circulating, which we know that for many of these vaccines, as an example, the messenger RNA vaccine has got an efficacy of about 95% against mild, moderate, and severe illness, but not from the due to the B1351 variant. So I'm going to share with you some data which some of you might have seen over the course of the past week. And this is a novel nanoparticle protein-based vaccine, which is produced by Novavax, which is a biotech company in the US. And I'm not going to go through the design of this vaccine, except to say, important of this vaccine is that it actually involves a pre-fusion spike protein, which is important in terms of which epitopes are visible to the immune system to mount an immune response to. And it's adjuvanted to a novel adjuvant known as matrix M. And it sort of formulates into a nanoparticle presenting itself to the immune system to induce the direct immune response, as opposed to messenger RNA vaccines or even the vector-based vaccines, which is basically using the codon, the genetic material coding for a protein for the cells to actually start producing the spike protein. So in the study, which was a randomized placebo-controlled trial, a phase 2 study, there were about 4,500 adults between 18 to 84 years of age that were enrolled in South Africa. About 240 were people living with HIV and 4,160 people without HIV. And the main primary objective of the study was to look at the efficacy of the vaccine against all severity PCR-confirmed COVID-19 illness, which occurred at least 7 days after the second dose. So this is a 2-dose schedule that is given 3 weeks apart with a window period of plus minus a few days, 2 doses, and we're wanting to see how well this vaccine works against all severity COVID illness. So what happened in the study, again, to mention inadvertently, is that we started enrolling in the study around about September. And as we continued enrolling, the last participant was actually enrolled on the 23rd of November. But as you can see from the top part of the slide, that shows you the different variants that were circulating in South Africa in different periods of the year. And as you see, around about September, probably the B1351 variant started to evolve and certainly started to gain much more traction by November. Now, the relevance of this is that most of our participants were actually enrolled, as I mentioned, between September and the 23rd of November, during which time very few of them would have contributed to primary endpoint cases because they needed to have received at least 2 doses of the vaccine. So it's only around about early November that participants would eventually start contributing to cases which would be useful for analysis in terms of the primary endpoint. And as you see at the bottom, essentially what ended up happening is that all of the cases that started to accrue were cases that were accruing when the B1351 variant became dominant. So as you can imagine, the majority of the endpoint cases were actually because of the B1351 variant. But just to pause in terms of the vaccine issue. Now, there's another big issue which has been discussed, and that is whether past infection actually protects against reinfection. Past infection with a prototype virus, something's gone wrong with the slide, so just bear with me, whether it actually protects against reinfection. What happened with the study is that we started enrolling after the first wave had passed. And what ended up happening is that 30%, almost 1 out of every 3 participants that were completely well, that volunteered to participate in the study, when we looked at a sample of blood that was taken at the time of enrollment, 30% of them showed evidence of past infection with SARS-CoV-2, which just shows how permissive infection was with the first wave of the epidemic. Unfortunately, what we then observed is that just looking at a placebo group and splitting it between those that had past infection and those that didn't have past infection, we showed that there was no difference in terms of subsequent all severity COVID illness. It was 3.9% in the placebo group stratified by previous seropositivity, which basically tells us that past infection with the earlier non-B1351 variant does not protect, unfortunately, at least against mild to moderate illness. Whether it protects against severe disease, we don't know, because there weren't many severe disease episodes in this particular study. So how did the vaccine perform in this particular study? Again, to emphasize that of the endpoint cases, of which we had 44 eventually, we had completed sequencing about 27 of them by the time we unblinded the study, and 93% of all of the endpoint cases, of all of the cases contributing to the primary analysis, were due to the B1351 variant. 93%, which is similar to what we're experiencing in terms of the percentage of people that are getting infected with the virus. Over 90% of that is from the B1351 variant. When we look at the efficacy of the vaccine against mild, moderate, severe illness restricted to HIV negative participants, we've got a point efficacy estimate of 60%. When we look at all participants, inclusive of the HIV positive group, of which there were very few, only 148, as you see at the bottom, basically the point efficacy estimate does drop to about 50%. So we can't say much as to whether this vaccine actually works in HIV-infected individuals. We can't say it does, and we can't say it doesn't. But what you can see is that in a vaccine group, the breakthrough number of infections in the HIV-infected group was 5.3% compared to 1.6% or 0.85% in the HIV negative group. So amongst the vaccine recipients, there seems to be more breakthrough cases in people living with HIV, and none of the other studies to date have actually done a stratified analysis. And this is important because even in the study, the HIV individuals that were enrolled were people that were on antiretrovirals and were virally suppressed. But despite that, we're seeing an increase in terms of number of breakthrough infections. Now, in tandem with the South African study, there was another study that was done in the UK, and in fact, the results were released at the same time. The difference being that this being a phase 3 study, the same dosing schedule involving 15,000 adults, including up to 84 years of age. And again, it was an endpoint-driven design. What we see in the UK study is 89% of all, the efficacy of the vaccine was 89%. And this efficacy was a mix of virus that was due to the 501YV1 variant and non-501YV1 variants. So roughly about 2/3 of the endpoint cases were due to the variant circulating in the UK. But before the most recent further mutation of this variant in the UK, as Penny had pointed out, essentially that variant remains fairly sensitive to the vaccine as well as naturally induced antibody immune responses. So it doesn't come as too much of a surprise that we've got a higher vaccine efficacy in the UK because it's dealing with the virus that hasn't mutated sufficiently relative to the mutations that has arisen in South Africa and Brazil, which would lend itself to increased resistance in terms of antibody-mediated activity. Now, what do we know about the other vaccines? So that is a single vaccine. So there's experiments that's been completed now with a Moderna and a Pfizer vaccine. And what you see with both of these vaccines is that there is significant drop-off in terms of neutralizing antibody activity when investigated against the variant, the B1351 variant. On the left-hand side, in the middle, you basically see sort of the early variants that were circulating, which had a D614 mutation. And then on the left-hand side, you see what is the relative neutralizing activity of the vaccine-induced antibody against the UK variant. And as you can see, there isn't much difference. There isn't much change in terms of the neutralizing activity to the UK variant as relative to the prototype virus, which is consistent with a high efficacy that you observed in the UK with the Novavax study. Unfortunately, what you see on the right-hand side, both for the Moderna as well as the Pfizer vaccine, is that there's between an 8.6 and a 6.5-fold reduction in terms of the sensitivity of the virus to this vaccine-induced antibody. So the virus is becoming more resistant or shows resistance to the antibody that has been induced by these messenger RNA vaccines. And the virus I'm referring to, obviously, is the South African variant. So what are the implications for other vaccines? We don't know. The bottom line is that right now we don't have a specific threshold of protection. So a minimum concentration of antibody that we know would protect the person against severe disease, or a minimum concentration of antibody that's required, functional antibody that's required to protect the person against mild to moderate illness. So it's difficult to tell exactly what the implications are in the absence of actually doing studies to see what is the clinical significance of this attenuation in terms of the clinical readouts we're getting from the laboratory. What we do know across the vaccines, although there are caveats in terms of these comparisons because they don't use exactly the same assay to do this sort of testing, we do know that some vaccines, as an example, the vector-based vaccines, are less immunogenic than the messenger RNA vaccines after 2 doses of vaccine. And in fact, the protein-based vaccine, the Novavax vaccine, performs even better than the messenger RNA vaccine. So there are differences across these vaccines in terms of the magnitude of immune response, the magnitude of the neutralizing antibody that is induced. So what happens with one vaccine might not necessarily mean that it would happen with another vaccine in terms of reduced efficacy or no efficacy. So I want to sort of end up with the last 2 slides, and that is just to reflect what we know of 2 vaccines. The AstraZeneca studies will probably be released from South Africa very soon. But what has been released is the Johnson & Johnson vaccine. And this is looking at the neutralizing activity in terms of against after induced by vaccine. So what I've highlighted in yellow is, the Johnson & Johnson vaccine study, which South Africa was part of, was a single-dose vaccine. And what I've highlighted in yellow is the relative neutralizing antibody concentrations for AstraZeneca vaccine and a Johnson & Johnson vaccine. And you see it's almost identical, roughly in a region where the GMTs of around about 200. So what we see with the Johnson & Johnson vaccine, which has been all over the news in the past week, is that it has a 54% efficacy against moderate to severe disease. There's no mention of mild infection here. The previous efficacy readouts that we've had is all about mild, moderate, or severe disease, but for Johnson & Johnson, they haven't as yet reported on mild infection. In addition to which, there's an 85% efficacy in South Africa against severe disease and death. And those confidence intervals are significant. So we don't know whether this vaccine protects against mild infection, and it might well not protect against mild infection. It might not reduce infectiousness, especially when the activity of the antibodies being sort of, in a sense, attenuated because of the mutant that's circulating. But what we do know is that it protects against severe disease, even in the context, and with Johnson & Johnson, all of the South African endpoint cases were due to the variant that's currently circulating. So in conclusion, what I've really tried to report, firstly, is that unfortunately for South Africa, past infection with the earlier virus that was circulating does not protect against reinfection with the N501V2 or the B1351 variant. We don't know whether it protects against severe disease due to this variant, but it doesn't protect against mild and moderate illness due to the variant. As I mentioned, the preliminary results of 2 separate studies on a spike protein, the nanoparticle spike protein vaccine, shows efficacy of 89% in the UK against a variant that against virus that included the variant that was circulating in the UK with the 501Y mutation. And for the same vaccine, I left out the vaccine efficacy here, as I've reported, that efficacy in people without HIV is roughly about 60%, but in a context where 90% of the endpoint cases were due to the B1351 variant. So this study was the first to report efficacy against the 501YV2 variant or a B1351 variant. And Johnson & Johnson's study basically, at least for severe disease, shows that it's able to protect against severe disease to this variant. The differences between the UK and South Africa possibly is because of differences in variant as well as differences in terms of socioeconomic conditions. And I think where the field is moving now is for the messenger RNA-based vaccines, for the vector-based vaccines, as well as for the protein-based vaccines, probably in the next few weeks, or at least the next few months, you're going to see a shift in terms of manufacture of bivalent vaccines, which are vaccines that will target both the earlier version as well as the variant that's currently circulating in South Africa, and would also be effective against the variant that's evolving in the UK as well as in Brazil. So obviously a number of acknowledgements for the number of volunteers and for the huge effort that's gone into the study from a number of different investigators across South Africa, and for funding and confidence of Novavax to do the study in South Africa. Thank you for your attention.
Thanks, Shabir, for that overview of the current variants and the vaccines that we currently have. So I think it's going to be an interesting discussion afterwards. But before we do that, I'm going to invite Charles to give his presentation. So Professor Charles Feldman is an emeritus professor in the Department of Internal Medicine at the Faculty of Health Sciences. His main interest has been in pulmonology and infectious diseases related to pneumonia, but he's also developed a sincere interest in ethics and ethics of research as well as vaccination. And so he's ideal to talk to us about, in the current era, about ethics and vaccination. Charles?
Okay, thanks very much, Praveen. Can you hear me and see my slides?
Yes.
Okay, so I've titled my talk, COVID-19, Coming to a Practice Near You. And my interest in ethics comes from me having been on the Human Research Ethics Committee of Wits for many, many years. So vaccines are obviously very important, with the exception, this is one quote, of safe water, no modality, and not even antibiotics has had a major effect on mortality reduction in humans. Another publication said that few would argue with Bill Gates, who said vaccination is the most effective and cost-effective tool ever invented. And of course, if we could have effective vaccines against what was called at that time, prior to COVID, the big three, malaria, HIV, and TB, it would have a major impact on human health. But vaccine development comes at a price that is not only financial, but also societal. So just to set the scene about medical ethics, the common idea is that the pillars or the principles of ethics, which we talk about as being due to Beauchamp and Childress, are the four that I'm showing there. Respect for autonomy, so the patient has the right to agree to or refuse treatment, or obviously vaccination or procedures or whatever else. Beneficence, and that's hopefully what we all do every day. We should act in the best interest of our patient. And then non-maleficence, which many say is one of the fundamental pillars of ethics or principles of ethics. First of all, or above all, do no harm. And lastly, we've got justice. So, for example, it refers to many different aspects, but including distributive justice, which is the distribution of scarce health resources and the decision of who gets what treatment fairly and equally. And in fact, vaccination has an impact on each and every one of these aspects of the principles of ethics. I do need to say to you, because I don't have a slide on it, that in general, both in ethical practice as well as research, as long as the potential benefit outweighs the perceived risk, so beneficence outweighs that of perceived or maleficence, that issue is likely to be ethical. So this is a slide I've developed to try and summarize some of the ethical issues, both with regard to pandemics and vaccination. So let's talk a little bit first about pandemics, and that has evolved into a term called pandethics, which is obviously pandemics and ethics. So we've had isolation and quarantine, lockdown, limitation of movements, limitation of the ability to buy things such as alcohol and smoking, and that obviously does impact against patient autonomy. We've had a big issue regarding resources, since we live in a country without unlimited resources, not that any country has unlimited resources, and this refers to justice or distributive justice. And one of the things we all know, and we've heard this many, many times, is that one of the things that COVID pandemic has highlighted and shown is the major impact on low socioeconomic circumstances, and this has been labeled the COVID inequalities. But let's go back to vaccines. So some of the issues, and they're more than this, I can't go through them all. What about ongoing placebo-controlled trials now that we do have some vaccines that have been found to be effective and have been registered at least for use in some areas? There's also, and I'm sure many of you have heard this from the president himself, what about global access to vaccines? Should it be distributed equally between developed world and developing world countries? And the talk that nationalized distribution or acquisition of vaccines on that basis rather than equally among all populations, maybe not ethical. Then again, who should be vaccinated first? And I'm going to talk to the issue only of whether the allocation should go primarily to participants in clinical trials who only got placebo. What do we do if people refuse vaccination? Because obviously enough people need to be vaccinated to achieve herd protection, as I prefer calling it. And then what about compulsory vaccination, particularly of healthcare workers? So let's talk about placebo-controlled trials. So in the situation, we know that the therapeutic use of a placebo involves intentional deception or incomplete disclosure. Because although we've got an information sheet, the patient still at the bottom line does not know whether they got the active ingredient or the placebo. And placebos relieve symptoms and improve conditions, many, many conditions. Now, in 1996, the Declaration of Helsinki, and we are signatory of the Declaration of Helsinki, declared that placebo-controlled trials can't be justified when there's an effective standard of care. The US refused to sign it, and they still do not believe or abide by the latest Declaration of Helsinki. So when you're thinking about a placebo-controlled trial, what you need to ask, the questions that need to be asked, is there equipoise with regards to the benefit-risk ratio? So do we know that the benefits are likely to outweigh the risks of the intervention of whatever that is? Are there effective alternative therapies? And clearly there are vaccines coming online that have shown efficacy, given what our previous speakers have said. There's also the issue of current standard of care. What is available in the country? If nothing is available, then a placebo-controlled trial may be ethical, because at least half those people are going to get an intervention that they otherwise would not be accessible to. And just to remind you that the Declaration of Helsinki actually revised the idea of placebo-controlled trials, and the stringent issues were compelling and scientifically sound methodological reasons for using a placebo, or in the case of minor conditions where the subject is not likely to be harmed. So there's been a lot of debate about should placebo-controlled trials of COVID vaccines continue. The WHO ad hoc expert group published in the NEJM, given I'd like to remind you that this was last year and things are changing. Ethics changes, the goalpost changes continuously. And what I say tonight may not necessarily be the same as what I would say tomorrow or in a week's time or in a month's time. But they have said, and remember, we do not have access to routine vaccination for COVID in South Africa at the moment. So that while vaccine access is limited, many available vaccines are still investigational. Our vaccines have been approved by SAPRA for use in clinical trials, but they've not been approved for routine use other than emergency use, as I've mentioned. And until the health recommendations have come out of health agencies that routine vaccination should occur, that placebo-controlled studies should continue. There obviously are a number of reasons for that. We need larger scale trials involving hundreds or thousands of cases to look for rare effects in the placebo group, rarer effects. We need to assess long-term efficacy, and most of the studies have been short-term. But of course, whatever I say is this is going to change because it may not reflect how we test vaccines going forward. The next issue is who should be vaccinated first. And I think many of you know that healthcare workers have been targeted, particularly frontline workers, for vaccination. Now, many may agree or disagree with it, but from an ethical point of view, it does make sense to vaccinate healthcare workers first. And I mean the range of healthcare workers. And this is described in ethics as the multiplier effect. For every healthcare worker whose life is saved by vaccination, that healthcare worker can go out and potentially save multiple patients with COVID. So the saving of one healthcare worker could contribute to the saving of a number of non-healthcare workers. But there has been a suggestion that maybe trial participants who've been given placebo should be first in line. They have contributed substantially to science. They have been aware of the circumstances of the vaccine trial, but they now know that vaccines are available. I mean, what, for example, about the ethics if everybody else in the country or the world is getting vaccination, having the same characteristics as the trial participant, but the trial participant may have been given placebo. But in order to determine that, we would need to unblind ongoing studies. And as per the publication, it says this may damage trials because unblinding for that reason may in fact be a protocol violation, but it also would prevent the ongoing efficacy assessment of many of those vaccines. Also, the fact that although numbers may be smaller, a number of the major vaccine studies that Shabir has mentioned recruited something like 30 to 40,000 cases. And in those cases, if we had to pool them, there would be a large number of individuals who received placebo who would have to be vaccinated if we prioritize the placebo group, which would defer equivalent vaccinations for frontline workers. Now, the third issue is that of Edward Jenner. So this is a sort of a biology of him. And unfortunately, the website that I show underneath, it's not accessible at the current time, which is a pity. Praveen saw what I said about that website, but I can't verify it at the moment. But he was born in the UK. He invented the first vaccination for smallpox called VACA, equals a cow. And that's where the word vaccine comes from. And in 1980, as a consequence of his original vaccination strategy, the world was declared free of smallpox. He's recognized as the founder of immunology, and his publication was the first publication in immunology. He was widely ridiculed. Critics claimed it was repulsive and ungodly to inoculate somebody with material from a diseased animal. There was a cartoon in 1802 showing that people who'd been vaccinated with cowpox or the equivalent sprouted cows' heads. But ultimately, the advantages of the vaccination and protectiveness won out, even among medical societies and so on, and vaccination soon became widespread. It continued, though, for a while, and in 1910, Osler, arguably the father of medicine, challenged the anti-vaccination movement to come with him, 10 of them, together with 10 vaccinated individuals, to go in to a smallpox outbreak area to deal with those patients. And he promised them that what he would do is treat them when they got sick and also arrange for their funerals once they died. And that's with Edward Jenner. Now, so that's the 18th century, fear and mistrust, but the anti-vaccination thinking receded in the 40s to 80s. There was a boom in vaccine science, discovery and manufacture. The public became aware of wide outbreaks, which were now being curtailed by vaccine and their desire to protect their children. There was a baby boom and increased education and wealth, and it receded. But it changed because multiple vaccines became developed. There were much fewer visible outbreaks to change people's minds. More vaccines were developed and added to the circumstances. And then the media came in, and the media allowed dissemination of poor science and anecdotal evidence of harm. And many countries, for example, dropped their DPT programs and suffered major escalations of pertussis. Now, in 1998, Wakefield published his publication based on science that was later recognized as being not exemplary, about the relationship between the measles, mumps, rubella vaccine and autism. So in a somewhat cynical frame, one author said, let's look at statistics and see what we could do, because you can show almost anything with statistics. So on the left vertical axis is the sales of organic foods that occurred over that time period. And if you look on the right-hand side, the individuals diagnosed with autism, and the conclusion that you could theoretically come to is that the eating and consumption of organic foods obviously is the cause of autism. And Wakefield's study was retracted from the Lancet. He was disbarred from further medical management of patients. And although a number of theories were forwarded as to how these use of the MMR vaccine could lead to autism, they've all been discounted, as have systematic reviews of this association. Now, what about mandatory vaccination? Do we need it? So in some countries, vaccination rates are very high. Outright opposition, if you look globally, is only around 2% of people in most countries. But there's a much bigger number of people who are so-called vaccine hesitant. And the speed of the COVID-19 vaccine development and approval has concerned many people, as one can see regularly in press. But of course, many of the vaccines are based on well-established vaccine development mechanisms and so on. And of course, many of them have gone through multiple trials. So it's been interesting that surveys in the UK showed that 70 to 80% of UK people would get the COVID vaccine if given to them, and 60 to 75% in the US, which is pretty high. Now, many of you will know that last week, the Center for Societal Change, UJ, and HSRC published research on South Africa's public willingness to take a vaccine. And you could see the bottom line, 67% would definitely or probably take a vaccine if available, 18% definitely or probably not, and 15% of adults didn't know. The bottom line is, how much do we need for herd protection? It's suggested 60 to 70% may be adequate. I've seen figures of 67%, which would be the group. But clearly we need to work on those cases who are uncertain whether they would take the vaccine or not. What about mandatory vaccination? It's controversial. It pits ethical principles of non-maleficence against autonomy. It's been endorsed by a number of medical societies and organizations, but it is a complex trade-off between personal autonomy, bodily integrity, and the public group. So what do we think is more acceptable? Mandatory lockdown or mandatory vaccine? Limitation of freedom of movement or consequential bodily interference, which is called the right to bodily activity, integrity. So I'm going to show this. It comes from a site called Vaccine Nation, which is very nice, and it shows the unbelievable impact of vaccination. And I'm just going to show one just for fun. In 1923, there were 1,560 cases of tetanus in the US. On the right-hand side, in 37 cases had tetanus or developed tetanus in the US because of vaccination in 2013. And in the middle, you could see that's exactly the same number of patients who died of lightning strikes. And there are many, many issues related to this. And my last slide is, in order to have a successful vaccination program, we need to have buy-in not only from the government, which we now seem to have, and healthcare professionals who are more easily convinced, and the public, but we need to ascribe a very important role to the media in making sure this is successful. So thank you very much for your attention.
Thank you, Charles, for that excellent overview on ethics of vaccination. So I think we're going to go on to some Q&As, and as the presenters have given their presentation, there have been some interesting questions in the chat box. So Charles, and in fact it applies to Shabir as well, from a question says, research ethics guidelines specify benefit sharing as an ethical obligation. Was there negotiation prior to starting trials for access to efficacious vaccines for communities, participants, and South Africa?
Yeah, I mean, I can try and address that. It's a very, very difficult issue. Those are components of not only international, but local regulations, such as good clinical practice in South Africa and so on, the Declaration of Helsinki. And I think it's important for local individuals like Shabir to have conducted these studies, because we need to see are they needed in South Africa? They clearly are, these vaccines. But we also need to see, and a lot of tonight's webinar has been, are they effective in the South African situation? Because if they're not, there's no point in getting them and registering them and administering them. So I think, yes, that is a very essential component of the ethics of vaccine studies. And it's something that is extremely important to make sure it's a study that is not only worthy for the country it's going to be done in, but those individuals are likely to have access to the vaccination.
Shabir, any comment?
Yeah, so I think it's an important question. And I think, firstly, I think we need to distinguish the role of investigators and the role of other role players. So within companies as well as within a country, there are different streams in terms of who does what and who has got what responsibility. In fact, it would be construed as an undue incentive for an investigator to negotiate with a company for preferential access should a vaccine be found to be effective in your own setting. The reason being that that could lend itself to investigators manipulating data to come out with a positive outcome, which you can't actually allow. So this is not a responsibility of investigators by any stretch of the imagination. There's an obligation on the part of companies to ensure that the products that have been evaluated in specific populations do become available in those countries. But it's sort of somewhat loose-ended in that it doesn't talk about pricing, it doesn't talk about anything else, it talks about the product becoming available. The prerogative of negotiating with companies in terms of access is the duty of government, of the people that are going to procure a vaccine. Where we were in South Africa is that we were in a highly favorable position in terms of doing these studies in South Africa, which government should have leveraged in terms of taking it as an opportunity to start early negotiations with companies and start entering into bilaterals, which didn't happen, unfortunately. So I think it's, yes, there is an ethical obligation of companies to make the vaccines available, but we need to be clear in terms of whose responsibility it is to negotiate such access. And that, for me, is the most important.
Just a brief additional thing from me. So one of the things our ethics committee does, so Wits' ethics committee, because that's what I speak for, our committee always says, what about post-trial access to the vaccines? And we always, as an ethics committee, push those companies to ensure that if the treatment or the vaccine is found to be effective, that it would then allow individuals who are on the trial, and particularly those individuals who are either got placebo or who are doing well with the vaccine, had post-trial access, so to speak.
All right, Charles. So I'm going to stick to the ethics theme a little bit, since there's a lot of questions. So I mean, you did mention about health workers. Firstly, should all health workers definitely be vaccinated, and what if someone refuses?
Yes, I think it's an ethical responsibility for all healthcare workers to be vaccinated. It's not only to their own benefit, but they are dealing with patients, some of whom are very vulnerable. For example, the elderly, those with comorbid illness. And if they get a mild infection, for example, and pass it on to their patients, that will be an ethical issue. So in many countries, mandatory vaccination of healthcare workers has been accepted. Mainly what I'm talking about is influenza, because this is prior to COVID, and I'm not sure of new regulations. But many countries have indicated that compulsory healthcare worker vaccination, for example, against influenza, is essential. In fact, many societies, infectious diseases, respiratory and others, have endorsed that. And many states, for example, in the United States, have had that as a mandatory requirement of licensing in that state. And many health institutions have required that as a condition of service. There are many things that we have to do as healthcare workers that we are required to do. And there's no reason why we shouldn't be required, other than if there's a medical issue or a serious religious, cultural, or ethnic issue for not getting vaccinated. In some circumstances, they've said, right, if you're a frontline healthcare worker and you won't get vaccinated, you need to be relieved of your duties there, and you could do other duties, maybe admin and so on. But in some situations, it has been a compulsory condition of service. And I think there's, from an ethical point of view, it does pit things like autonomy against beneficence and non-maleficence. But from an ethical point of view, we could justify it in the protection of the patients we look after. Above all, first of all, do no harm.
Thanks, Charles. So one of the questions asked of me in this week is saying, well, the AstraZeneca has now arrived in South Africa and the health workers have been prioritized. So firstly, we're going to get the first dose, and we've only got about 1.5 million doses. So Shabir, how long can we wait, because the AstraZeneca is a 2-dose regimen, before we get the second dose? And do we still continue the mitigating factors, that means masking, social distancing, if I do get the vaccine with the first dose?
Yeah, so in terms of the last part, those mitigating factors are going to need to remain in place probably for the rest of 2021 at least, including amongst healthcare workers that are vaccinated. Remember, when you are vaccinated, the efficacy readout is basically providing risk reduction at the population level rather than at an individual level. So when you've got efficacy, as an example for AstraZeneca, which is 70% against the non-B1351 variant, it doesn't mean that you as an individual have got a 70% lower risk. At a population level, it is, but not at an individual level. So you can't second guess as to which individual is going to be protected and where the breakthrough infections might take place. So I think we just need to be careful in terms of contextualizing that sort of data. So in terms of the spacing, so the UK obviously has gone with the first dose and follow up with 3 months later. And there's a number of factors that play into that. Firstly, it's with regard to availability of vaccine, as well as trying to get saturation as quickly as possible in terms of vaccine coverage. For the AstraZeneca vaccine, as similar to the Johnson & Johnson vaccine, you see efficacy evolving within 14 days after the first dose. So it's not that you need 2 doses of vaccine to get protection. Just with a single dose, you're already getting protection as early as 14 days. And if you do have supply constraints, it's better to try to maximize your coverage rather than to get 2 doses in as quick as possible. Paradoxically, it actually assists in terms of delaying the second dose, in terms of immune responses. So if you delay the second dose, you actually get a better immune response than with more narrow intervals. So a second dose at 8 weeks is better than a second dose at 4 weeks, and a second dose at 12 weeks, in fact, is better than a second dose at 8 weeks in terms of the magnitude of the immune response, which probably is important in terms of durability of immune response. The third part to the answer is that it also depends where you are in terms of the epidemic curve. So where we are currently is we're very much on a downward descent. We're on a descent in terms of the current wave. And in 2-3 weeks across the country, we probably would have plateaued with a resurgence. So the risk of infection also starts diminishing, which means that you again got more opportunity to delay your second dose, to spread out the vaccine more broadly to start off with, but in addition to that, to actually get a better immune response. So I think for AstraZeneca vaccine, it makes perfect sense to actually space it to as late as 12 weeks later. The other vaccines don't necessarily apply the same principles because we don't know about whether extending the interval between the messenger RNA vaccines, as an example, would result in a heightened immune response.
Right. Thanks, Toby. The one thing that's impressed me, despite the results of both the J&J as well as the AstraZeneca vaccine results in South Africa, so we appear to have a lower immunity response as compared to the other countries. But what has impressed me most is still that the vaccine, even in our current environment where the variants are rampant, the number of severe infections is significantly reduced.
And I think that's a really important issue. And it might well, so remember the current data from AstraZeneca doesn't speak to efficacy against the B1351 variant. All of the analysis, including what was released today in the Lancet, really speaks to non-B1351 variant. We probably will publish data on the AstraZeneca vaccine shortly, which will provide an efficacy readout for the B1351 variant. But what you mentioned is critical. So the Johnson & Johnson vaccine, in terms of the immune responses that it induces, and Penny's probably busy with experiments as we speak, in all likelihood, you're going to see knockout of antibody activity against the variant in the lab. Yet that vaccine is reducing severe disease by 85% against the variant. So there's something that's happening. Now, obviously, we know that immunity induced by vaccination is not just about antibody, there's also T-cell immunity. And the mediators of immune responses that you require to protect against lower airway infection, which is your severe disease, are probably very different than what you require to protect against upper airway infection. And most of your mild to moderate illness falls in the spectrum of upper airway infections and respiratory infections rather than lower airway. So it might be that CD8 lymphocytes are being primed to be much more potent in terms of its activity in the lung, and the antibody is not being that great in terms of protecting against upper respiratory infections. But I think there is this, and I think it's important, when we see the AstraZeneca results, we need to keep this at the back of the mind. Because if we end up showing in the South African study, we've had a very young population in AstraZeneca study, we only had mild and moderate infections, and it might well be that the vaccine doesn't work against that spectrum, but that doesn't dismiss biologically that the vaccine could still protect against severe disease despite not having efficacy against mild infection.
Thanks, Sabir. So this is one for Penny. Penny, so one of the questions is, some scientists have expected the punch of this virus will grow weaker over time, and which is in fact what happened with other viruses. In fact, certainly watching the influenza virus in 1918, it appears that the pandemic panned out because the virus just became weaker. Is it possible that this virus could become weaker?
I think when we think about viruses becoming weaker, we're not talking about a matter of months or even years. We're talking about eons. Immediately what the virus is going to do is what all viruses always do, is it's going to select mutations that are naturally arising, and it's going to pick the ones that deal with the immune response it's seeing. And at the moment, what we know from many studies across South Africa and many other countries is that the immune response to SARS-CoV-2 is very focused and very similar between people. It hits one little spot or maybe two little spots on SARS-CoV-2. So it's a relatively easy thing for the virus to escape. So I think that's completely unsurprising. There have been really detailed, elegant studies understanding at the monoclonal antibody level how the immune system sees the virus. And in many cases, it uses the same germline antibody genes. It's a very, very narrow response. And that makes this early escape very easy to explain. I also think that, viruses rapidly select mutations that increase transmissibility. And so that's also unsurprising that we are seeing that increased transmissibility leads to increased spread. And, it's not a surprising thing that we're seeing the same mutations coming up again in all three of those lineages. So I hope that over many, many years, whoever asked that question is right, but it's not going to happen in a matter of weeks or months, unfortunately.
So in the meantime, we're going to focus on trying to make vaccines that on epitopes of the virus that in fact provide this increased transmissibility. And do we target a single epitope or multiple epitopes?
Yeah, Shabir already alluded to the fact that various companies are already moving very fast to incorporate the South African variant and other variants presumably into their vaccines. And I think what's really changed over the last year for SARS-CoV-2 vaccine design is we have really powerful tools that allow this kind of plug and play approach to vaccine design. So we're better placed than we ever were before to be able to slot in new variants, new mutations. But I think another crucial thing that we need to understand, at the moment, we're only looking forward. We're trying to understand whether people previously infected with the old variant are going to be susceptible to the new variant. We need to understand the reverse as well. We need to understand whether there's commonalities between the two variants that inform vaccine design that can tell us where better sites of vulnerability are in the virus, particularly on the spike. And we need to incorporate those into vaccine design. And again, we have a history of doing this in lots of other viruses. I suspect it sounds all very good and easy. I suspect, and Shabir's laughing at already, and I suspect we're going to find that it's going to be as easy for the virus to escape the immune responses elicited by the next vaccine. And who knows, maybe it'll go back to the old sequence. So we're going to have to keep on top of it from a genomics point of view. We're going to have to understand which way it goes, and we're going to have to look for better targets than RBD, which is clearly too easy for the virus to get away from.
So thanks, Penny. I mean, one of the questions that was put to me earlier as well was that, I mean, we all know the story about the vaccinations that we've received here, and the next batch coming in some part of the second quarter, which we don't know. But should be basically saying to get herd immunity, we need to be vaccinating close to 40 million people. And with a vaccine that is currently available, that is 50 to 60% effective, will herd immunity really be effective in reality in South Africa, at least not in 2021, is the question posed.
Yeah, so the calculus around herd immunity has just changed since this variant has evolved. In fact, you don't need to vaccinate 60% or 67%. With this more transmissible virus, you probably need to get immunity around about 80% of the population needs to develop immunity for that herd immunity to evolve. And when talking of vaccine and herd immunity, it's not just about a percentage of people that are vaccinated. Your percentage of community that develops immunity is a product of the efficacy of the vaccine against infectiousness and the coverage. So a vaccine that has only got 10% efficacy, as an example, against infectiousness, even if you've got 67% coverage, at a population level, you only got about 10% protection in terms of transmission. So the sort of single magic figure doesn't exist. In addition to which is compounded in that you're not going to get to herd immunity just with vaccination. For some things, for smallpox as an example, yes, we did. For polio, we did. But there's a very different dynamic in terms of transmission with those viruses. In terms of SARS-CoV-2, right now, given what we've seen in the efficacy trials and what we're observing with the variant, I think reaching herd immunity as a global community is unlikely to occur anytime soon, even if we had all of the vaccines available, the current generation of vaccines available. There's too much immune evasion with the variant to basically get adequate extent of community immunity. So it's going to be probably a product of both vaccine-induced immunity, provided that vaccine actually reduces infectiousness, coupled with natural acquired immunity. So it's not just a product of coverage and vaccine-induced immunity, there's also the natural immunity that's induced. Unfortunately, I think a few weeks ago, I was shouting from the hilltop that we need to vaccinate to get to herd immunity. Now I can realistically tell you that we're not going to get to herd immunity anytime soon through vaccination, through vaccines, certainly not through the current generation of vaccines. But you mentioned 1918, and 1918 was more than just the virus mutating to become less virulent. In fact, the driver behind 1918, the second wave ended up being more severe than the first wave because the virus actually mutated and became more transmissible and more virulent. So there were many more people that died in the second wave. But essentially over time, what happened is that there was sufficient immunity developing across the community in the absence of vaccine, where we weren't able to get rid of the virus, but there was sufficient immunity to try to sort of reduce the frequency and the magnitude of these waves. And that is what we're heading probably for SARS-CoV-2. COVID-19 is not going to go away in our lifetime for all intents and purposes. It's here to stay. But it's an issue about how we try to minimize the frequency of the resurgences and the magnitude of the resurgence. So we're really in a space now, in my mind, where we are now, we need to recalibrate, stop believing we're going to get rid of this virus, which was never going to happen, and start learning how to live with the virus. But at the same time, trying to protect those that are most vulnerable from developing severe disease and dying.
Right. So I mean, so the idea that we're going to get rid of masks and social distancing is not going to be. That's going to be with us for at least another few years. Correct.
I wouldn't say a few years. I'm a bit more optimistic than that.
Okay.
I think, I mean, again, this virus is completely unpredictable. In 6 weeks from now, when Penny discovers there's a new virus with a different set of variants, then we'll have a different discussion. I think that probably for the rest of 2021, we're in a difficult space. And then depending on the next generation of vaccines and how effective they're against the variant, as well as in terms of the type of coverage we can get, hopefully by early 2022, we should be in a reasonably good space to get back to some level of normalcy. But it's still a long road, and there's a lot of uncertainties, unfortunately.
So I've been asked by a couple of patients who are immunocompromised to say, and they fear that they are at a much higher risk of developing severe disease. There aren't that many of them randomized in clinical trials. So what's the recommendation for them receiving vaccines?
Yeah, so like I said, they need to get vaccinated. High-risk individuals need to get vaccinated as quickly as possible with the current generation of vaccines. There's no reason to believe that you're going to run into problems if better vaccines become available, especially with a different construct, and that your immune response will be poorer to the other vaccines, especially when they're using a different construct. So in the context of the data that came out from J&J, which basically shows 85% reduction against severe disease and death. And that is essentially because they enriched the study population for the elderly and those with comorbidities. That itself is extremely important, extremely good reason for high-risk individuals to get vaccinated as soon as possible. Even if these vaccines don't show efficacy against mild to moderate illness, if they become available, and if you're at risk of developing severe disease, I wouldn't think twice about being vaccinated. You're not going to cause any harm. We know the vaccines are safe. The vaccine might not work, but you're not going to be causing harm, and you might derive protection against severe disease.
And what's the current recommendation for pregnant women? Because that's a recurring question as well.
So unfortunately, at this stage, for the messenger RNA vaccines, WHO recommends that pregnant women that are at high risk of being infected receive the messenger RNA vaccine. There's no similar recommendation for the vector-based vaccine, the AstraZeneca vaccine. The studies in pregnant women are currently underway for multiple vaccines. So right now, if you are at high risk, and if you've got access to the messenger RNA vaccines, that is recommended for use. And probably in the next 2 weeks, WHO will make a recommendation for vector-based vaccines in pregnant women.
So the one other question that I got asked in the change room the other day was, they weren't quite sure that if we got vaccinated as healthcare workers in the next few weeks, there was not clarity about when the next batch of vaccines will arrive. Would it be possible to change with the second dose beyond 12 weeks to another vaccine? Because from my understanding, we were now going to get the Pfizer vaccine next.
Yeah, so those sort of interchangeability studies haven't been done. And I think it might depend on a platform that's been used, what technology has been used. As an example, if you start off with the AstraZeneca vaccine, there's good reason to believe that you actually will get a better booster response if you then take the Johnson & Johnson vaccine as a booster dose. So you got this heterologous type of combination, and the Sputnik 5 is an example. They start off with adeno 26, and then they've got an Ad5 vaccine as a booster. And you actually have less interference by having this heterologous type of boost. But in terms of across platforms, by starting with vector-based and then going to messenger RNA, that data, theoretically, it should work because all that you're doing is you're presenting more protein, spike protein to the immune system to boost the response. So theoretically, it should work, but those studies haven't been done. But in the eventuality that that does occur, transpire that you start off with AstraZeneca and there's no more AstraZeneca vaccine, I mean, those studies are in fact underway in the UK. They are natural experiments where exactly the same thing is happening, where individuals that were participants in a study basically are now receiving other vaccines, and they're actually looking at how they, what sort of immune responses they do mount. So we will have results fairly soon, probably sooner than when the second vaccine comes to South Africa.
All right, so Charles, in the last few minutes, I mean, there's been a suggestion in some countries who've suggested that those who refuse vaccination, I mean, they won't get forced, but if they do get infected, they may be denied ICU admission. Is that ethical?
So the feeling is, and let me start, that many of the patients we look after have got lifestyle issues that are responsible for the medical conditions that we have to look after. Individuals who smoke with respiratory diseases and so on. And it is not ethical to go and refuse. I mean, if you think about it, people with lack of exercise, people with diet, inappropriate diet, people who smoke, people who take excess alcohol. So many of our patients who we have to look after have got issues that could have been prevented by doing things differently. So I think the idea with ICU, and you're right, there is one of the countries, one of the individuals on the ethics council of that country have suggested that what we do is not, people who refuse vaccine should carry a card that says, I refused vaccine. And they should also then agree that if they get ill, they are not going to have the ability to be admitted to ICU and so on. Now, from the ICU point of view, obviously we do triage patients. We would take patients who've got a better chance of recovery with conditions that are reversible and so on. But the idea of just excluding individuals from, say, ICU admission on the basis of age or the basis of underlying comorbid conditions or whatever is really the slippery slope. And many people believe that that is unethical, that it shouldn't be on that basis. If there is, you know, a stress on beds and one has to then decide who comes into the ICU or not, then a whole lot of issues are taken into account. Among them, age, comorbid conditions, likelihood of survival, the underlying diseases, the current condition, and so on. But to just exclude patients on the basis of one thing, such as age or comorbid conditions or lack of vaccination and so on, I don't think would be seen to be ethical. If there is a stress on beds, if there's a resource issue, I think that it needs to be, many things need to be taken into account. But as I've indicated, many patients who end up in ICU have got lots of conditions that could have been prevented if they had done things differently in their life. So no, I think that's an opinion. Ethics changes all the time. Often different ethicists have differing opinions, but I would think excluding those patients just on the basis that they would not have the vaccine would not be an ethical issue.
Since Charles, you write a lot on vaccination with influenza. What's your recommendation with regards to vaccinating for the next flu season?
Well, it depends on who you're talking to. So you're talking to an individual who's probably had more, as an adult, who's probably had more vaccines than any other adult I know for various reasons, which we don't have time to go into. I believe in vaccines. I have vaccines. I have the flu vaccine every year. I'm sure Shabir and maybe even Penny could answer this better than I could. But the idea is that it's good to be vaccinated. It prevents you. I'm in the high-risk group from a point of view of age and comorbidities and so on. And I think it's absolutely important. But even as a healthcare worker, I can understand the ethical issues in making, for example, vaccination of healthcare workers mandatory. I mean, the one reason to do it is to protect yourself. But the other issue is non-maleficence, do no harm. And the ethical description that's often given, if you're a healthcare worker, many of them are young, otherwise healthy, they may get influenza, it may be relatively mild, they've got a work ethic, so they don't stay home, they come to work. And the story goes, so what happens if you have a stem cell bone marrow transplant patient in an intensive care unit with the costs involved, who now gets nosocomial influenza from the healthcare workers? I mean, that is a considerable ethical issue. So it depends on what circumstances you're talking about, but I personally can find no reason not to have yearly influenza vaccination. And I can tell you that my whole family, I'm talking about myself and wife, children, and grandchildren, have yearly influenza vaccination.
Thanks, John. I mean, there's compelling science behind that recommendation, correct?
No, there is, and Shabir will tell you that even in pregnant women, it's very, very important. I saw the realities, for example, even with H1N1, when one of our registrars who was pregnant developed H1N1, and you know, and there was an issue of, you know, what should happen there. So yeah, I think there are many compelling reasons and many compelling issues. But even if you don't have those issues, there's absolutely no reason not to be vaccinated. I mean, Shabir and Penny may want to answer this better than I can, but if you've been vaccinated, sure, there's waning of immune responses and so on. But with the next pandemic, the influenza pandemic that comes out, if you've been previously vaccinated and the virus is obviously a mutated virus, but a similar one to what you had before, there may be some degree of protection. I can see no reason other than medical reasons where it's contraindicated, and there are very few of those, that an individual shouldn't be vaccinated against influenza every year.
Thanks, Shabir and Penny. Any comments on that one? Flu vaccination this winter?
Yeah, I mean, yeah, I agree. The short answer is I agree with Charles. When a vaccine becomes available, get a vaccine. There's a much longer answer, an academic answer and discussion in terms of why we didn't experience a flu season last year. And in all likelihood, the season this year might well be very attenuated as well, or it might be more severe than the past. But I think that's a discussion for another day. But despite all of the limitations around flu vaccine, I think when there's a good match to the circulating virus, it has got high efficacy. And as Charles pointed out, especially WHO itself indicates that your priority group for vaccination, firstly and foremost, are pregnant women, and then individuals at high risk. But at the population level for healthcare workers, it just makes sense to use a vaccine that's available and that is safe.
Penny?
Nothing more to add.
Okay. All right, so I think we're running out of time. So just to Shabir and Penny, give me a kind of a, you know, there's kind of a lot of doom and gloom out there about the pandemic and the vaccines and the variants. Give me a kind of a silver lining view about this whole story.
They've worked to some extent against the variant. I mean, I think that that's very reassuring. I mean, you know, the Novavax still showed activity against the variant, and I think there was a lot of fear that it might not. Many of the other vaccines come in with much higher titers than convalescent plasma. So in many cases, we're doing better. We've seen there's a drop-off in titers, but we don't know what's enough to protect. And certainly, as Shabir's already alluded to, for J&J, those low titers somehow protect against disease. So there must be other arms of the immune system at work that we don't yet understand. So I think there's a huge amount still we have to learn about these vaccines and about how they work. But no, I don't think we should be totally doom and gloom. I think the fact that they showed some efficacy, it's certainly a setback. And as Shabir already mentioned, we're in a different place to where we were 2 months ago, I guess, 3 months ago. But, you know, they're still showing some activity and some, you know, so we have a lot to learn.
Yeah, I fully agree with you, because I was really, when I saw the 56% result with the J&J vaccine, but when I saw that it actually prevented in a very high percentage of patients, over 80%, severe disease, and it means you in fact in preventing hospitalization, that was a real silver lining for me. Shabir?
Yeah, so I guess my silver lining is we got much more knowledge about this virus than 12 months ago. So that's a good starting point to start combating the virus. And as you mentioned, we might not have the perfect vaccine for COVID-19, but I think consistently across all of the vaccines, the efficacy against severe disease is usually higher than efficacy against mild to moderate illness. And that applies for the messenger RNA, comes in at 95% for all severity. But when you look at hospitalization and death, it goes to 100%. And obviously there's overlap of the confidence intervals, but there's a consistent picture emerging, which is that these vaccines certainly, irrespective of the variant or not, still reduces the risk of severe disease substantially. So it might not be the vaccines that we were hoping for to be able to get back to a normal life as quickly as possible, but I think what we're wanting right now is to minimize death from COVID-19 and to reduce the pressure on our healthcare facilities. So even with this first generation vaccine, despite what limitations they might come with, and I'm fairly confident that in a very short period of time, probably by the middle of this year, we'll have one of the first bivalent vaccines that becomes available. But whether that's going to do the trick or whether we're going to chase after another variant is anyone's guess. But I think there's reason to be hopeful in that we do have something that now protects people from dying protects people, at least reducing the risk of dying from COVID-19.
Thanks, Shabir. So unfortunately, I think we have to end the webinar now. And we were not able to answer all the questions that were posed by the audience. But what we'll do is in the next issue of the journal, we'll try and publish the responses to some of the outstanding questions. So we'll collate them and then I'll send them to the participants and maybe we can reply that way. So before I close, and on behalf of the editorial team at Wits Journal, I extend my sincere appreciation to the panelists, Professor Marty, Professor Penny Moore, and Prof. Feldman, who have graciously given up their time and educated us and informed us on the latest thoughts and evidence regarding vaccination for COVID-19 in South Africa. This is clearly a moving target, and we hope that we may be able to host another webinar in the near future. So a special just thanks to Andrew Joseph from Wits Press, who helped coordinate this webinar, and he certainly made my task much, much easier. My thanks also to our sponsor this evening, The Forge, and those at Forge, Molela Sele, Ryan Hollyball, and Richard Pitthouse. And thanks to Victoria Hume and Rita Kruger for promotion of the webinar. And finally, thanks to Kathy Naidu from Sebinet. So my final thoughts are, remember, the mutations have not changed the basic nature of the virus. The spread can still be inhibited by the same common sense measures, and I'm sure Shabir will agree with it, masking and social distancing. So to all of you, good evening, and I hope you all stay safe. Thank you very much.
Thanks.
Thank you.
Thank you.